So our next speaker is Dr. Leif Andersson, and he will be introduced by Dr. Max Rothschild. Max.
Well, good morning, everyone. It's a pleasure to be here today, and I certainly want to thank the organizers, and in particular, Lauren, for inviting me to make a few comments today. And before I introduce our speaker, our distinguished speaker, I just want to comment on why I'm attending this seminar. Jim Womack was an extraordinary person, a colleague of mine, and a friend for over 30 years. It might be interesting to how I actually met Jim. In the early days, some time ago, I was a quantitative geneticist. And in fact, people have talked about their connections with other people here. Harris Lewin was an undergraduate. I was his TA in quantitative genetics at Cornell University. So you only have me to blame if he has trouble understanding certain things. And I went from quantitative genetics, and I made a difficult choice in those days, and I decided I was going to get interested in individual genes, and I did work in the MHC, and then I kind of wandered into gene mapping and in particular comparative work, looking at the pig, which of course in Iowa where I was working was the most important species, just like cattle are important here in Texas. And I did some work initially with using human gene probes to actually make discoveries in the pig. And my movement into gene mapping, I then met Jim and a number of the people in this room. Those were the heady days of gene mapping. You know, everyone was at some level being friends and yet competitors. There wasn't much money in those days. And all the techniques were new. Every week it seemed like we had a new set of markers, a new way to proceed. And Jim was a great guy to talk to about all of this because he was well grounded in the comparative side. Jim and I worked side by side together as genome coordinators. He was the beef genome coordinator and I was the swine coordinator for over 20 years. Jim was always collegial, helpful in our professional work, and most of all, a friendly and caring individual. It's already been mentioned that Jim was an avid fisherman. Anybody who knows me knows that I'm an avid fisherman as well. In fact, I think the hardest thing about coming to Texas and to A&M this time was I'm not fishing. Lauren invited me to come fishing, but I came with my wife and she frowns on me fishing more than 3 or 4 days a week, so that wasn't allowed. But almost every visit I came here to A&M, I always fished it with Jim. And in our conversations, even though they started out professionally, most of the conversations led either to discussing football, basketball, or fishing. And we shared pictures. Often he showed me his big fish or one of his grandkids' fish that they had caught, and I would share mine with him. And I actually, one of my big regrets in life, in my fishing part of my life, is that I never got Jim to come to Canada with me and fish with me. Jim and his wife were great hosts, and they hosted my brother and I on several fishing trips here in College Station. And on the last fishing trip I took with my aging father and my brother was here at Jim's ranch. The kindness and the friendship of the Womacks was really special. And while I think it's important that we all remember Jim's contributions to science, I think it's him as a person that really made the relationship special. And I'll always remember that kindness and friendship, and I'll cherish that forever. Well, let me just make a few comments about our speaker, who I've been asked to introduce, because that's the important part here too. Leif Andersson, I've known him and collaborated with him for a long time, especially in the early days of the PigMap Consortium. I was the member of the American lab that joined the European Consortium, and Leif was kind enough to share materials. We used one of his mapping families for a number of our early mapping discoveries. Leif is a professor here at VMBS and a fellow of the Hagler Institute for Advanced Study. He holds professorships in functional genomics in the Department of Medical Biochemistry and Microbiology at Uppsala, and in molecular animal genetics at the Swedish University of Agricultural Sciences. Leif's mostly known for his leadership and his discovery work in the areas of genomics and molecular study of domestic animals. He published one of the first, I think maybe the very first animal QTL study that everyone went on to use as a model for future work. He's been particularly effective in using the pig as a model organism, but he's looked at many other organisms as well. Leif has many honors. He's a member of the National Academy of Sciences, the Royal Swedish Academy of Sciences, and he's won the Wolf Prize among many of his awards. So let's welcome Leif to the podium, please.
Okay, it's great to be here. So I wanted to start my presentation with this picture, which is taken almost exactly 2 years ago, 2023. And I'm very grateful to Lauren and his wife Mary, who organized this so we could visit with Jim and Raby on their farm. And I've known Jim since 40 years. So he came to Uppsala, I think 1983, just when he decided to start working with cattle genomics. So in our department, we had one famous cytogeneticist, Ingmar Gustafsson, who had discovered a lot of translocations and so on. And Jim wanted to know about the cytogenetics in cattle when he started his work. And since then, I've been interacting many times with Jim. And this was a lovely day. And this is his farm in March. This is the most beautiful time to visit Texas, I think, because of the temperature, which is like Swedish summer, and also because of the flowering, which is out and so on. So that was the reason why I took the initiative to have this trip yesterday, which was really lovely. Okay, so I plan to talk about. So the thing is that I've been working, my first 30 years of my career, I worked on domestic animals. And the next 30 years, I'm working on natural populations. So I think it's fair to divide the talk between domestic animals and natural populations. And my passion in science is the link between genetic variation and phenotypic variation. That is what always have been what I would like to study. So I would like to compare what we have found in domesticated animals and then go over to the natural populations and see how that refers to each other. So this is many of the species that I've been working on, so not only pigs. So I've worked on all domestic animals, basically. Rabbit is not here at the moment, but we worked on those as well. And of course, much of the diversity that you see in these pictures are Mendelian traits that is easy to map and identify the causal mutations. And that's what we have been doing over time and led to exciting stories, I think, and so on. And the reason why we have this is that humans have been cherry-picking mutations during the domestication. Domestication happened like 10,000 years ago. And since then, we have always cherry-picked variants that have been appealing to us. And these are examples of that. But this is, as I said, for me, genetic maps and assemblies are just the tools to go to the genotype-phenotype relationship. So when we started to do gene mapping in pigs, I wanted to create a pedigree that could tell us something about how have we transformed a wild species into a domesticated species. And what you can say is that this project was in fact inspired by Darwin. It's because this picture you have to the right there is from Charles Darwin's book, Phenotypic Variation in Plants and Animals Under Domestication, which was published 1868, 9 years after the original species. And this was in fact research that Darwin did to provide support for the theory of evolution by natural selection. So what he did, he compared wild and domesticated plants and animals and to see how have they changed. And in this picture, he compared the wild boar with the domestic pigs. And I got this vision that now is when we start to be able to do mapping, now is the time to go to the next step and use molecular genetics to try to find out what are these changes. And what you see in this picture is the segregation of coat color, which we basically have solved most of this. The only thing left to solve is why some pigs, an interesting aspect here is these black spots here are somatic mutations that happens in every generation. And what we haven't solved yet is the difference between these which are red with black spots and white with black spots. But I hope to solve that also in some time. But I wanted to talk about a large effect mutation that we discovered. So Max mentioned this first QTL mapping, where we did QTL mapping because there's this big difference in phenotype, and that is shown on the previous picture. So what is quite interesting on this picture is that you see that the domestic pig here is really fat, and that is the pig in the mid-1800s in England. And then the selection goal was to make pigs as fat as possible, because you needed the energy to do work in agriculture and industry. But in this case, which is 150 years later, it's the opposite. Then we have selected for leanness. After Second World War, the main goal was to select pigs that produce protein and not fat. So when we crossed these guys, what we noticed was that the hybrids became more fat than the domestic pigs. So, and we mapped then a Mayer QTL to the IGF-2 locus, which has a huge effect on meat in ham and the amount of fat and also the size of the heart. And you see the amount of variance explained for muscularity is 30% of the variance is explained by a single locus, which is enormous for a quantitative trait. And interestingly, Michel's group mapped the same locus in a cross between Pietran and large white as we mapped in wild boar versus large white. And together we had a wonderful collaboration where we were able to nail down this to a single base change in an intron of IGF-2. And here come to comparative genomics. And if you, so we mapped it to this locus, and we had in fact genetic data that nailed down a single base change. And then we did a comparison to other species. And what is really fascinating is that so this is an intron, it's a CPG island in an intron of IGF-2. So you see all the CGs here in red. And then you compare with cattle, horse, dog, rabbit, mouse, human, rat. And what you see is that you have 16 base pair here, which is conserved among all these species, except the domestic pig, which has an A at that CPG. Today you have the Xenomia project, and there's more than 100 mammals that has a sequence here. So if you add another 100 mammals, you will see that this is super conserved among all placental mammals. I don't think there's any example that we know that this site around here has been mutated in any mammal. So this was the story. So what we were able to show was that, so this is the mutation. So wild boar, Chinese pigs has the wild type, and many of the breeds in Western countries used for meat production has this single base change. We also showed that this mutation knocks out the binding of a nuclear protein in a transcription factor. This was the first time ever anyone went from a complex trait down to a single base change in a non-coding region. Has not been done in humans, has not been done in mouse, not in Drosophila, not in C. elegans, but it was done for the first time in pigs, 2002, far before we had an assembly. This was rejected by science because the third reviewer said, you need to tell us what is this protein here, which was what I thought was a ridiculous request, but we submitted to Nature and they published it, so we were okay. So But the story doesn't end there. So this is from the 2003 paper. And this is the yell shift experiment that we do. So this beautiful thing, you take the oligo from the wild boar and it binds a transcription factor and you take it and you take an oligo corresponding to the sequence from the domestic pig. It does not bind. So the protein is there, but in a yell shift experiment, there's so little protein, so there's no way that you can find out what it is. The thing is that at that time, 2003, there was no technology, efficient technology to pull out this transcription factor, but that was developed. So 6 years later, we were able to use the mass spectrometry to fish out the protein binding to the wild type sequence, but not to the mutant sequence. And it turns out that it was a previously unknown protein. You could say that it was the dark side of the genome 2003 or 2009, because when the human genome was annotated, they missed this protein, because the protein is sitting in an intron of another gene. So this is ZC311A, which occur in all vertebrates. But in mammals, there is a gene sitting in this intron. And if you look at sequence conservation, you could immediately see that this is something important sitting in that intron, but it was missed because they annotated. And the reason why they miss it is that this is a domesticated DNA transposon that integrated in the gene of mammals more than 200 million years ago. So this Hermes is the same family of DNA transposon present in the housefly, but what happened in the mammals is that this DNA binding domain has been duplicated. So this is the evolutionary history. So it integrated before the split between marsupials and eutherians, but it's pseudoionized in marsupials and platypus, but is present in all placental mammals. So it's one of the innovations that happened during the evolution of the placental mammals. And if you look at the DNA binding domain, so when we did that, there was not a single missense mutations in the DNA binding domain, so extremely well conserved. So then we did, in the first paper, when we described this, we also did ChIP-seq and identified. So we confirmed this is the IGF-2 in mouse, and this is the binding site. And we could, based on all the sequences, this is the consensus sequence motive. So if you now use the Sonomia data and take a look on this, then you find a lot of these binding sites in the placental genome. And what you see here is that this is a Philo-P score. So the majority of these is around 0. So I would say that in this ChIP-seq experiment, the majority is false positives. But then you have this long tail, and it turns that if you rank the CBIT6 binding motifs, how well conserved they are among all placental mammals, IGF-2 is ranked as number 67. If you do the same thing in human, you see the same thing. You have most of these sites, because this is overlapping data, of course, but then you have this long tail of CBIT6 binding site in IGF-2. So apparently it's really well conserved, but in human there are about 300 genes that has CBIT6 binding sites that are evolutionarily conserved. So what we decided to do was to study the importance of this system. So we generated both. So these are 4 types of mice. So this is the wild type, but then we knocked in the pig mutation on a mouse. And then we knocked out CBIT6, and then we combined the two. So we had mice that was both had a knock-in on IGF-2 and knockout of CBIT6, and then we compared them. So this was published 2018. So it took almost 30 years from we started cross until we had this data. So sometimes you have to be persistent in science too. So we get these mice, and you look at this and you get a little bit disappointed, right? They look quite similar. It's the same picture though, but you couldn't see a phenotype. So So this is what I think is truly fascinating. You know, this site is conserved, look like it's conserved in all placental mammals, and you knock it out and you don't see any obvious phenotype. But what I would argue is that this interaction between CBD6 and IGF-2 is essential for fitness in a placental mammal. And what I think this brings to the placental mammals is metabolic flexibility. And that is illustrated with the phenotypes we observe. So if you look at muscle development, you see that, so we looked at 4 types of muscle, both red muscles and white fibers. And in each case, you see a significant effect of this. So you increase muscle growth exactly as we see in the pig. So this is the reason why this mutation is so widespread in pig. And interestingly, you see the same thing by knocking in the IGF-2 mutation or knocking out CBD6. So this is, so apparently this has this huge effect on muscle development. But we also see that the transgenic mice get larger kidneys, larger heart, larger liver. So our conclusion from that is that this locus, so that if you compare with pig, we also, you mentioned that it affects both muscle and the size of the heart, and that is replicating in the mouse. But we didn't see an effect of kidney or liver in the pig system, so some differences between placental mammals. The circulating levels of IGF-2 are dramatically increased if you compare, and you see the same thing with these 3 models. So remember now, so the first one here, that is the knock-in, this is the CBD6 knockout, and this is the combining the 2. So the conclusions from the CBD6 IGF-2 story is this is an important mechanism regulating muscle growth and growth of internal organs in placental mammals. And you could say the phenotypic effects are modest compared with this huge difference in IGF-2 levels. And I think if you look into the IGF-2, it has a very complicated regulation with a number of binding proteins that is regulating the effect of increased IGF-2 expression. And when we look at the Sonomia data, it turns out that this region is one of the most well-conserved non-coding regions in the genome of placental mammals. So I think this is a classical effect of a large effect mutation, which have been under very strong selection during pig breeding. So most of the pig meat you have eaten, if you eat pig, then is come from pigs having this mutation. So I used to say this is part of our wealth. So each pig that has this mutation has about 4 kilograms more muscle at slaughter and less fat. So it's an enormous value each year, this single base change. However, it's very clear that most quantity traits in domestic animals have a highly polygenic background. And my lesson for that was I collaborated with Paul Siegel at Virginia Polytechnic Institute who developed these selection lines. So he started 1957. I was not involved, as I was 3 years old. But he started with a set of inbred chicken, crossed them to break up linkage, and then he started to select them. And he selected, and after 52 generations of selection, he had a 13-fold difference in body size at 8 weeks. So only selected on body size, and a lot of metabolic shifts between these appetite changes and so on. So I thought it was a great resource to do linkage mapping. So we generated a cross between those. And we published this 2005, and the conclusion was that there was no large effect QTLs in this cross. So instead we see, so this is the chicken genome and the significance for, this is the estimated additive effect. And the interesting thing with this curve, I think, is that if you look at this estimated additive effects, is that across the genome, you almost always are on the side that the allele from the fast-growing chicken is increasing growth. So that fits really with the pseudo-infinitesimal model that there are many loci, each with a small effect. And if you do fine mapping, this, what we call peaks here, often break down into smaller peaks. So here he starts with a quite narrow genetic background, do selection, and get enormous response, but it's highly polygenic. So we had problem to publish this because we couldn't really highlight any specific genes at the time, So that was not considered so interesting at the time. But after that, thousands of human jivas has given the same message, right? That you take a trait and you see this highly polygenic effect. But this was 5 years before the human jiva started to come out. So another thing I would like to mention from the work I've been doing is that my view is that large effect mutations, they exist, but as large effect on the positive selection or directional selection is rare, but large effect haplotypes are more common. And that is what I call evolution of alleles, haplotypes, or supergenes. So what happens in nature and also in domestic animals, alleles may increase fitness by accumulating favorable mutations. And my favorite example is evolution of dominant white color in pigs, but it's not the only example. So although the history of the animal domestication is quite young, it's 10,000 years, we have several examples where we see multiple mutations accumulating on the, say, alleles or haplotypes. So this is the story about, so we start with a wild boar, and the first mutation which happened is a duplication, 450 kb duplication, and including the KIT gene, and that gives the patch phenotype as you see on this, which was from the previous pictures I showed you. KIT is a very important tyrosine kinase receptor, which has a critical role for migration of melanocytes out to the skin. And KIT is one of those genes. You have one megabase, only one gene, but the region is full of long-range regulatory elements that affect the expression of KIT. Because KIT is very pleiotropic. It occurs in germ cells. It occurs in hematopoietic cells. So if you knock out KIT, you kill the animal because this is essential for hematopoietic development. But it's also critical for pigment cell development. Then there's next mutation hit, and that's a splice mutation, and that splice mutation knocks out the tyrosine kinase domain. So then you have two copies of the gene. One copy is, you have first a regulatory variant, then you have a structural change that knocks out signaling. So you have a signaling dead receptor. And that, so if you only had a splice mutation, that would be recessive lethal. But since you have the duplication first, this become viable and with a very strong phenotype. So this is what I call a fitness landscape for adaptive haplotypes. So you can't go with one mutation from this peak to that peak. And you have first a duplication, then you have splice mutation. There's also a small duplication that also contribute to the phenotype. And the interesting thing that this is the allele so far described. There are hundreds of alleles in mouse. No allele has such a strong effect on pigmentation as this mutation has. And it's because it's a combination of several mutations. Okay, let's shift to natural populations. So just to give you a feeling for the species. So Atlantic herring is one of the most abundant vertebrates on Earth. And so what I used to say is that, if you would like to study how natural selection affects the population, then what you should do is you take an infinite population, split it into multiple subpopulations that are also infinite, and then you expose them to different environmental factors. The herring is as close as you can come to that situation. So multiple subpopulation, but all of enormous size. So the total population size is about 10 to the 12 to 1 trillion individuals when they're spawning. So I argue that I think Atlantic is a gold mine for genetic studies. So one thing, it's a fish. So why is that important? I think you see much more ecological adaptation in a fish than what you do in a bird or in a mammal. And the reason is that birds and mammals have found a way to control environment during development, because we do it at a constant temperature. But the fish is exposed to the environment to a completely different way. The herring is subdivided into subpopulations that show ecological adaptation, salinity, temperature, light conditions, and so on. And this huge population size, and also there's gene flow between, so the populations have a homing behavior, but there is some gene flow, and that eliminates genetic differentiation at neutral loci. So when you see this, what the presentation I will do now about genetic differentiation, then you should remember that for most genes you can't distinguish a fish from the Baltic Sea or from the American coast, on the American East Coast. It's a demersal spawner, so what that means, it puts the stick eggs on the bottom, which means that the eggs are sitting at the place of spawning for the whole development period, about 2 weeks or so. And that means that facilitates homing behavior and make homing behavior to increase fitness, because it allows ecological adaptation. High fecundity, so each female can make like 30,000 eggs. And it could breed for up to 20 times during their lifetime. And another very important reason is that it is recently, within the last 8,000 years, adapted to a new environment, the brackish Baltic Sea. So 10,000 years ago, we still have glaciation where we are in Sweden and the Baltic Sea. So then the Baltic Sea opens up and the herring comes in and adapts to this environment. It's good to eat, in fact. So why is that important? Well, it's important because for the last 100 years or so, herring is one of the 10 most important fishes in marine fishery, if you look at the biomass collected. And it's been extremely important for food security in Northern Europe. But that means that fishery biologists have been studying this for a long time. And what our results has led to a revolution in the way how you do stock assessment, because we have identified the markers to distinguish the different stocks. So now tens of thousands of herring will be genotyped each year with a SNP chip we have developed. So within 10 years, we will have an enormous data set where we could look at the frequencies from year to year. We could see how recruitment was based on environmental conditions and so on. So they will help us to generate data, which is great. So then is the question, so how polygenic is ecological adaptation in a species with a huge breeding population, about 1 trillion individuals? So in each generation, each base is mutated. The herring test each single nucleotide in each generation. So the infinitesimal model for polygenic trait loci, it suggests that polygenic trait like human height is controlled by an infinitesimal number of loci, each with a tiny effect. A very important prediction is that if you have a selection process, you should not see any major shift in allele frequency, because each locus has such a tiny effect. So you could get a huge effect by shifting the allele frequency at many loci. And that is what you can see, in fact, if you have in systems. So for instance, we argue that about the rabbit domestication, that we rather saw shifts in allele frequency rather than fixed. Very few variants are fixed in the domesticated rabbit. So we argue that is a very good indication of a highly polygenic model. So what happens if we do this? So this is what it looks like when you compare Atlantic with Baltic herring. So this is using 6 million markers. So remember now that most markers are down here, but then you have this, so each dot is one single SNP. So you see this huge beautiful signals so what you could say is it's highly polygenic for sure, but it's a major deviation from the infinitesimal model, because you shouldn't see this type of shifts in allele frequencies. So we just give one example, and that is rhodopsin, which is one of the genes that has responded to selection. So this is, if you zoom in on that chromosome 4, So this is, you heard about Manhattan plot, right? This is the Uppsala plot. We all have one high building, this is a cathedral. So, and then you see that rhodopsin is just at the peak here. And then now we just look at the SNPs that is unique to the Baltic Sea, which we don't find in the Atlantic. And now you see the sweep. So you see this here, these blue ones here, which are the most significant ones. So you see this beautiful, so the signal goes away from the peak. And that is because of recombination. Or based on the recombination, we could predict how long time it took to basically fix this variant in the Baltic Sea. And it required, in order to see this, because if it goes very slow, the peak will be very, very sharp. But here we see the sweep signal. And the selection coefficient must have been very large to get this signal. And interestingly, there are two, so we have a missense mutation up here, residue 261, and we have one here. So what is interesting is if you see this sweep here, and then this is the only SNP that comes up in this, like a second phase of selection. We think both of them, this is natural selection. So, as a geneticist, so you have, this is the Baltic allele has tyrosine at this position, and in Atlantic allele you have phenylalanine. So if you're a geneticist, you would say, well, and in fact, if you align all fish species, you will see that all fish has either tyrosine or phenylalanine position. So you could say it's a tolerated polymorphism. But if you're a protein chemist, you say, well, hydroxyl is quite interesting because they could do interesting things in proteins. And then you look, where is this residue located? Well, it's located here, and this is retinol, where light absorption takes place. And this hydroxyl molecule is pointing like a finger towards retinol. And what you can predict is that this mutation, so this is machine learning method that has been developed by a group in California. And then they've taken all rhodopsin molecules, which has been characterized with regard to absorption. And then you could predict that this shift here is causing a shift of 8 nanometers here. This other, this is the mutation which has, this has both, and that gives the same. So we think this mutation was selected because it shifted the light absorption. This other mutation has shifted for another reason. It could be stability of the protein when you have shifted this first. And if you look, so this is satellite imaging of the Atlantic and in the Baltic, and you could see how red shifted the Baltic Sea is, which means that herring with this shift in rhodopsin see better in the Baltic, and outside in the Atlantic we don't see, so white here means 100% Atlantic, and this is 100% Baltic, and some populations here are completely fixed for this variant. But what is fascinating is that if you then start to look at other species, it turns out that 1/3 of all fish living in freshwater or brackish water has exactly the same mutation as the herring has. So at least 20 times the same mutation have happened when species have adapted to freshwater and I think that for me was a surprising finding, that a single amino acid change can have such a profound effect on ecological adaptation in a natural population. So this is convergent evolution. So this is another example of a large effect mutation. So we find them also in natural populations, and this that we have 2 mutations accumulating on the same protein is another example of this evolution of alleles, as I mentioned with the pig. So let us go to another trait, and now we talk about genetic regulator of reproduction. So an interesting thing with the herring is that it has 2 types, 2 strategies with regard to spawning, spring or autumn. So what they basically are doing is utilizing different, because the key for for herring is that they spawn, so they hit when there's abundant food. So they are using like different resources for the survival of the larvae. And then in this, so this is a new Manhattan plot, and what you should notice here, so here we compare populations in the Baltic Sea, and here is populations in Atlantic Ocean, so completely independent Jiva studies. And what you could see is that the most important loci is replicating. So you have particularly this locus here, which we have TSHR here. But you have others. So for instance, this is something that is selected in the Baltic amongst in this contrast, but not in the Atlantic. So that locus should not have anything to do for setting reproduction. It's probably a gene that is important for spring or autumn spawners, where there is a difference, but the Baltic Sea is a different environment. Let's zoom in on this chromosome 15. So in fact, that looks like one single signal when you look at all chromosomes. But when you zoom in on this chromosome, what you could see is that it's much more, it's multiple peaks. So this again, this graph is for population from the Baltic Sea, and these are populations from the Atlantic Ocean. And so we have a number of interesting genes. So this is SOX11B, which sits on this. It's just upstream of SOX11B. You have TSHR, calmodulin. You have estrogen receptor beta as a signal. And look how well this replicates. You can count 1, 2, 3, 4, 5, maybe 6, 7 loci. And if you then compare in the Atlantic, 1, 2, 3, 4, 5, 6, 7 loci. So perfect replication. Have you ever seen a human jivas that replicate so perfectly when you take two independent populations? I don't know, maybe Michelle knows. So what I would say here is that, so this is what I think is a supergene, in fact. But it's a supergene that do not contain inversion. So what we see within population, we see very strong LD in this region, at least among these here. So if you don't have an inversion, then you have more recommendations. So you could, so I would say that this is inherited as a haplotype block in populations, but the most important factors are here, which are kept together by natural selection. So this is illustrated by, so this is an old split between these haplotypes. So here is Atlantic herring, and this is Pacific herring. So this split we have dated to about 3 million years ago. But look at the split between this is the spring spawners, and this is the autumn spawner haplotypes. And so you see, it's not 3 million, but in the order of 1 million years has this been separated and evolved over time and accumulated changes. What you also could see here is that there have been a selective sweep among the spring spawners. They have much shorter branch lengths. So something happened in the spring spawners that led to this sweep signal. There's one blue here, so that is in fact, so what we have done here is that we have collected spring spawners, autumn spawners, done haplotype phasing, and made a tree. So one of the spring spawners was heterozygous for an autumn allele. So we know that there's gene flow between them, so they're not completely isolated. So once again, at neutral loci, we can't distinguish them. But at those loci that is important for reproduction, there they essentially fixed for different alleles. So once again, so evolution of haplotypes. So what this is photoperiod regulation of reproduction. And in all vertebrates, there is this system that sends day length and use that to time reproduction. So different species is mating at different seasons in order for the offspring to be born when there are optimal conditions. And so in human or mammals, we notice light through the retina, and then there is this, signaling to melatonin, and that leads to release of TSH, thyroid stimulating hormone. And a key factor in this pathway is activation of the TSH receptor. Birds have a similar thing, but in birds you could also get light directly into the brain because the skull is not as dense as it is in mammals. Same thing in fish. So we know what this region in the fish brain that is regulating this. And the fact that the most significant locus we have identified in this is TSHR, tells us that we are in this pathway. And luckily, I've recently got a new Mayer grant for fish biology experiment and high-resolution single-cell transcriptomics. So what we plan to do is to collect this region from spring spawners, autumn spawners, every month over the year to see how regulation is, how is it possible to delay the start of reproduction in one versus, because the day length is the same for what they experience. But so we think this could be an opportunity to really make a breakthrough in understanding how vertebrates set reproduction in relation to day length. How I'm doing with time. Okay. Yeah, maybe I show this, then we're done. So, so one of the things we've been working on is fact from a thesis from, so there's one thesis from Texas A&M University about herring, and that is Minal Yamsadekar presented that a year ago approximately. And one of the papers is about the MHC. So several in the audience have studied MHC, the most polymorphic gene in vertebrates. And we wanted to know, so if you now go to the most abundant vertebrate on Earth, so how does the MHC look like? And what you could see here is that this is the rate of synonymous changes. This is the rate of non-synonymous changes. This is maybe the most polymorphic locus in the human genome together with HLA-DQ beta 1. But you see the polymorphism in human MHC is small compared with the herring. So herring has higher DS, which means deeper ancestry, and it has much higher frequency of non-synonymous changes. So the herring MHC is much more polymorphic than the most polymorphic locus in the human genome, both because they have more copy number variation, they have more polymorphic genes, and there's a higher diversity between the copies. So this is like what I think is the beauty with the system, where natural selection is free to operate and develop the polymorphism. Last picture. So here we have taken AlphaFold, taken the structure, the protein sequence we have determined for the herring. Then we have added the sequence polymorphism of each residue and asked the question, where is the polymorphism located in this? And it turns out that red is polymorphism. So this is, with AlphaFold, you get this MHC structure, and what you can see is that all the polymorphisms, the most polymorphic site is sitting on the alpha helix or beta strand, pointing towards the peptide binding site. That's all. Thank you.
We have a few minutes for questions.
Yeah, 10. Take the microphone, Lisa.
So just sort of thinking loudly in a way, but so you're talking about these highly divergent haplotypes which you find in herring populations, and now you sort of point towards selection as an underlying explanation. But I'm just thinking, if you have a very, very large population size, so and if you look at the coalescent tree, for you sort of expect the most recent common ancestor to be very, very far, just because the time to fixation is a function of the, and half of the time to coalescence is due to the last coalescent. So half the time between now and the most recent common ancestor is the two last branches of your coalescence that come together. So I just wonder how much of what you observed in terms of divergence of some haplotypes could just be a reflection of neutrality, but a very large population size.
The argument here is that so what we see outside this region is that LD break down very quickly. So LD, if you're only a few hundred base pair apart, you see very little LD. So to see this, so what we see is these long haplotypes is only at some of these loci under strong selection. So, yeah. So otherwise they break down. And I think what I showed you on the TSHR is that I think you have like a type of supergene, but it tends to break down because of recombination when you don't have an inversion that really takes. So we published a paper last year. We have 4 inversions that supergenes that is really important for ecological adaptation in the herring. And what we see even there, we see there's a lot of exchange. These are also million-year-old, so they be going on for a long time. But what we see is that over time there is also recombination going on between the inversion copies, and that is what you see in Drosophila as well. So the inversion is not completely absent of recombination, but it's highly reduced. So,
Leif, you have the most beautiful examples of GWAS all the way down to the functional allele.
yeah.
Of course, in the human disease world, where we have hundreds of GWASs that don't succeed,
Yeah.
where is the missing heritability?
What do you mean, in human or
In human.
Well, I mean, there's a lot of things to discuss about that, and what I would say is that, in fact, I had the next slide here, so this is when we do GWAS in Darwin's finches, and also here we see very large. So we compared the Darwin's finches with human stature. So the beak size of Darwin's finches is about, the heritability is about the same as in human stature, 70% heritability approximately. In humans you have 1,000 genes, which explains about 35% of the variance. In Darwin's finches we have 6 loci, which explains 45% of the variance. So very different genetic architecture. But what I think is both herring and so what I think you have in humans is you have a lot of stabilizing selection. So that means that and also what I think you have is that you have slightly deleterious mutations that contribute to variation in these phenotypes, but they explain very tiny fraction of the variation. In the Darwin's finches and in the herring, we have had some very strong directional selection. So Darwin's finches evolved from a single species with the last million years. So this is the most important locus explaining variation beak size among Darwin's finches. And I think that has played a really important role for the Darwin's finches to utilize different food resources. So I think the evolutionary history of humans and these species is very different. So even there's people done the same studies on great tits in Europe, and they don't see this huge effect as we see. And I think there you also have a stabilizing selection, because there's great tits in Europe, they are competing with many other species. But on the Galapagos, these species come in, and there's a lot of open niches that they should expand into to utilize different food resources. So this locus, which allowed them to quickly change from one food resource to another, has been under strong selection. And then I could show this picture also, that this is a supergene as well. But this is a supergene which is not associated with an inversion. So I don't have time to explain all things, but this is the region, and it contains 3 genes, LMD3, WIF1, this one, and this locus HMA2, which is important for the story.